new discovery shows human cells can write RNA sequences into DNA. Cells contain machinery that duplicates DNA into a new set that goes into a newly formed cell. That same class of machines, called polymerases, also build RNA messages, which are like notes copied from the central DNA repository of recipes, so they can be read more efficiently into proteins. But polymerases were thought to only work in one direction, DNA into DNA or RNA. This prevents RNA messages from being rewritten back into the master recipe book of genomic DNA. Now, Thomas Jefferson University researchers provide the first evidence that RNA segments can be written back into DNA, which potentially challenges the central dogma in biology and could have wide implications affecting many fields of biology. This work opens the door to many other studies that will help us understand the significance of having a mechanism for converting RNA messages into DNA in our own cells, says Richard Pomerantz, Ph.D., Associate Professor of Biochemistry and Molecular Biology at Thomas Jefferson University. For example, this finding suggests that RNA messages can be used as templates for repairing or rewriting genomic DNA. The work was published June 11 in the journal Science Advances. Like theta, HIV reverse transcriptase acts as a DNA polymerase, but can also bind RNA and read RNA back into a DNA strand. The group collaborated with Xiaojiang S. Chen's lab at USC and used X-ray crystallography to define the structure and found that this molecule was able to change shape in order to accommodate the more bulky RNA molecule, a feat unique among polymerases. Our research suggests that polymerase theta's main function is to act as a reverse transcriptase, says Dr. Pomerantz. In healthy cells, the purpose of this molecule may be toward RNA-mediated DNA repair. In unhealthy cells, such as cancer cells, polymerase theta is highly expressed and promotes cancer cell growth and drug resistance. It will be exciting to further understand how polymerase theta's activity on RNA contributes to DNA repair and cancer cell proliferation.